so much. Thank you, Dennis, for that invitation. And before I even get going, and this is something I don't want to forget, I'd like to thank the NSF, EVO-DIVO, and plant genome programs, and the USD and IFA for supporting all of the research I'm going to talk about today. I hope that I can convey to the Kaplan Memorial Lecture Committee what a great honor you do to me by actually inviting me to do this lecture. So I knew Don somewhere in sort of this phase, but not quite, I mean, of course, I knew him all the way to the end. So I started graduate work at UC Berkeley in 1985, and fresh off the boat, Don was a little scary, but our relationship evolved along the way. I was trained in classical botany, a lot of anatomy, and I thought I knew everything I needed to know, but I did take his plant morphology course, and I can tell you that it really changed how I view plants and everything that I've done in my research after that. Don often used to say that everybody does very careful research, and you interpret your results, but whatever you leave behind that's really going to be important is how you make people think. So if you can change how people think, then that's the influence, that's the most important. And I think that it's going to be quite evident to this audience that he really did influence how people think. So Don said, and I'm sure you all know, that the leaf, within the leaf lies the true Proteus. From first to last, the plant is nothing but leaf. And so it's no surprise that he thought a lot about leaves, and he worked a lot on leaves. This is actually a picture from one of his papers describing all the different leaf types found in the angiosperms, and is also a really great segue into my talk, where I want to really focus on the two different kinds of leaves that angiosperms make. These leaves that have a single unit of blade or are simple, and then these other types of leaves that actually have multiple units of blade or are complex. So I really wanted to try to figure out developmentally and genetically how these different leaf forms arose. This kind of variation in leaf morphology is often found between closely related species, here illustrated in the genus Pelargonium. But sometimes you can actually find this kind of variation in response to environmental signals on the same plant. So simple or compound leaves in Roripa aquatica can be determined by many, many factors, including, as demonstrated here, by temperature. In addition to this evolutionary variation, developmental variation, heterophylous variation, which is environmentally triggered, there is also this aspect called heteroblasty, which is major morphological change seen from juvenile to adult vegetative development. And here it's best illustrated in this slide that Scott Pothig sent me 2 days ago when he heard that I was going to come here and do this lecture. He said, "You need this slide." So this is an acacia plant growing in his greenhouse at UPenn, and here you can see that the acacia has complex leaves in its juvenile form, and as it goes into adult morphology, it gradually transitions into these simple leaves. And so this process is called heteroblasty. So this is my, go back to home slide, and I'm going to really talk about the regulation of compound leaf development, which is very gene-based in the beginning, and then I'll talk to you a little bit about heterophily and then heteroblasty, and then try to compare these two programs to figure out what is common between them. So there was this idea, or two schools of thought, if you will, where Arber and other people who followed that school of thought said the leaf is a partial shoot, and it has an inherent urge towards the development of whole shoot characteristics. And in contrast, there were the German morphologists, and perhaps this is best articulated by Kaplan, who said the main difference between simple and compound leaves is that the blade region is subdivided into subunits or leaflets. So within that framework, we actually started to, in the beginning, just use an approach of candidate genes. And so we said, well, we should then look at genes that play a role in determining indeterminacy in shoots, and then we should also look at leaves that play a role in blade development, genes that play a role in blade development. and then frame this question as, are compound leaves partially determinate, or are they really simple leaves with lamina subdivisions? And this is really not an either-or answer, as you will see, and I'm sure you fully realize the answer lies somewhere in between these two extreme scenarios. So I first want to start talking about some of the genes that we looked at in order to understand compound leaf development and evolution. And so there's many factors, and we have focused on tomato as a model system because leaves can really be varied by a variety of factors. I'm going to go through some of these in the talk, but for now I will focus on developmental stage and phylogeny to look at leaf development. And so the genetic framework that was being used at that time was to think of shoot indeterminacy genes. I'll talk about two specific ones, and then also to talk about genes that regulate blade formation. So the first gene that we focused on was this knotted-like homeobox gene class, because we knew that these genes regulate shoot indeterminacy. When the shoot meristemnus gene, which is sort of the flagship gene in dicots, is mutated, then you can see that this Arabidopsis plant does not make a shoot meristem and does not produce any more leaves. The expression of these genes also differs between simple and compound apices, and the expression levels regulate leaf complexity. So I'm going to show you some of those data very quickly. So here in an Arabidopsis shoot apex, you can see that gene expression is focused on the meristem, and it actually turns off as the next leaf is going to form, and then it's off. And then in tomato, which makes compound leaves, and this is not Arabidopsis, but simply centered. And so in tomato, two different Knox genes actually have expression in the meristem and also in the developing leaf primordia. And then, as very nicely shown by Miltos Siontis and Angela Hay, when you reduce the expression of Knox genes in Cardamine hirsuta, which this is the natural state of the leaf, you reduce leaf complexity, and if you upregulate Knox expression, then you increase leaf complexity. So this seemed like a really good group of genes to look at for their role in leaf development. So then many years ago, Geeta Bharathan then did a broad survey of leaves throughout vascular plants, looking at the expression of Knox genes in the apices that bore these leaves. And the net result of Geeta's analysis was that every time the leaf was compound, there was expression of these genes in the developing leaf primordia. So this was very nice, but this was really a candidate gene approach, which was trying to imply things simply based on gene expression. And we wanted to try to move beyond that and look in a more sort of uninformed way and see what comes up if we just try to understand the causal basis using genetic mechanisms. So we tried to look at the variation in the Galapagos tomatoes, Solanum galapagense and Solanum cheesemania, which are really quite varied. And this is a much less complex leaf than the galapagense leaf. And it was known from genetics done by Charlie Rick that this was determined by a single semi-dominant locus. So we as in Seiske Kimura and Dane Koenig, who are in my lab, did a chromosome walk to clone this natural variation locus, and they found that this was also a Knox gene. And so the causal mutation was a single base pair indel. The gene was very interesting in that it was unlike regular Knox genes that have a protein interaction and DNA binding domain. This just had a protein-protein interaction domain, and it was lacking the homea domain. So then in order to understand how leaf complexity was being regulated by this truncated Knox gene, we then had to identify interacting partners. So this is published work, so I'm going through it quickly. And we showed that the tomato Knox genes interact with a bell gene in tomato, and so this was actually causal for another mutation that was known in tomato at the time called bipineta or BIP. And then we showed that Knox and BIP can partner each other, but if you express PTS in the mix, it can disrupt this interaction because PTS preferentially interacts with BIP. And this frees up the Knox genes in this complex and then allows for more compound leaves to arise. So that was the story for Knox genes. There's more to that. Every time I've tried to escape the Knox genes in whatever way I will, the data drags me back there. So, I won't go on and show you more data on those lines. So I want to move to this other question that we had, which was, is leaf complexity always accompanied by Knox expression? So, is this always true? And so, of course, in our head, this was always going to be true until we looked at this pea leaf out there. And so this was, again, work done by Gita. And she showed that in pea, there's actually no expression in the developing leaf primordia. A little before that, actually, work done in Julie Hofer's lab and John Innes had shown that leafy might be regulating leaf complexity in peas. So just a little aside on leafy, in Arabidopsis, leafy mutants have aberrant flowers, but their leaves are pretty normal. And leafy is supposed to regulate floral complexity or inflorescence determinacy, if you will. So Julie showed that in pea, when leafy is mutated, the flowers are aberrant, as you see in Arabidopsis, but the leaves become simple. So Connie Champagne then did a survey of the legumes and showed that, here's pea, but if you look in Lotus japonicus, there's actually very nice expression of Knox genes in leaf primordia, and in fact, she went on to do a survey of throughout the legumes at selected genera, and showed that in this clade, which is marked by an inverted repeat in the chloroplast genome, there's no Knox expression in leaf primordia, but throughout these basal groups, there is. And so I'm just going to show you some of that data, where in Mimosa and Acacia, Lotus, and Phaseolus, there's expression, but then, it doesn't matter how compound the leaf is, like wisteria, pretty compound. There's no expression, nor in Metacarda. So the derived clade marked by the inverted repeat in the chloroplast genome lacks Knox expression in leaf primordia. And P, of course, shows that leafy is important for making this leaf complexity. So then the question we asked was, what is the role of leafy in species where Knox genes are involved in regulating leaf complexity? And for that, we turned to tomato again and looked at a leafy mutation in tomato. So this mutation is called falsiflora in tomato, and here you can see that the flowers are quite like those pea flowers you saw, where they proliferate and they do not make the right floral organs. But for us, it was more important to actually look at leaf complexity. And what Connie showed was that while the primary leaflet numbers do not change, so here we're looking at wild-type falsiflora heterozygotes and falsiflora homozygotes, the secondary and intercalary leaflet numbers do change quite a bit. So this suggests that even in this group where Knox is a major player, there is a role for leafy in regulating leaf complexity. And we showed similar results in soybean by making RNAi of leafy in those plants. So then the conclusion from this study in the legumes was that in the pea clade, it's leafy that's the major regulator of leaf complexity, but that in most higher plants, leafy still has some role in regulating leaf complexity. So with this look at the indeterminacy genes, the shoot and floral indeterminacy genes, I want to turn now to the leaf fractionation or blade outgrowth genes. So the first gene I want to mention was published in 2008, and this was the group of genes known as NAM, standing for no apical meristem, or cup-shaped cotyledon genes. And so this group that first author was Blind et al. They showed, and I think Mintos was involved in this work. They showed that in diverse dicot clades, you always have expression of this cup-shaped cotyledons gene at the points that would separate leaflet primordia from each other. And then in several cases, they were able to either get mutants or do RNAi to downregulate cup-shaped cotyledon, and so this result is from Cardamini from that same paper, to show that when you have a certain level of complexity, by downregulating cup-shaped cotyledons, you can reduce leaf complexity and you can reduce leaflet lobing. So this gene seems to be quite important for the regulation of leaf complexity in cardamom and in most dicots. So I want to show you a few other leaf players, and I will move on to this one gene which we were quite enamored with for a while, and we should, I think, get back to it because I think it's quite fantastic. So the gene is called Fantastica, and it is big domain transcription factor, and it seems to regulate adaxial sulfates in leaves. Several years ago, when the gene was first described in Antirrhinum and named by Andrew Hudson, Andrew proposed in a very visionary paper that when you have adaxial and abaxial sulfates coming together, that's what allows the blade to grow out. So we looked at tomato Fantastica, and I'm just going to summarize a whole lot of work in just a few slides. We showed that FAN regulates adaxial identity in tomato, and so this is the normal tomato leaf, and this was work done by Min-Sung Kim, who was then a graduate student in my lab, and now he's a professor at Manchester University. Min-Sung showed that if you overexpress this gene in tomato, you can get very large simple leaves. So this is a tomato leaf, not a cabbage leaf, not a kale leaf. The greenhouse stock would not believe, but sometimes it makes tomatoes. In addition, by overexpressing FAN, ectopic blades can be made. So here you can see that this gap between a leaflet there and there has been taken over by blades that just kind of grew out. Sometimes when ectopic adaxial foci appear, then you can get these opposing leaf blades. And basically every piece of data we got really supported the hypothesis that Andrew had proposed, that juxtaposition of two different sulfates causes blade outgrowth. We also were able to actually use the gene expression data and some morphological data to define two different kinds of palmic compound leaves, and I thought that was a nice ancillary benefit from this work. So if you look at tomato, this is the expression of Fantastica, this is the adaxial domain, and you can see that adaxial domain running down the center of the leaf and the rachis as leaflets are being made. So we propose that this green adaxial domain, when it encounters the orange adaxial domain, leaflets grow out. But if you can reduce the levels of these by antisense experiments in our case, then if you get just a patch of FAN expression, you can make a cup-shaped blade. And then in order to take it to its extreme, I want to show you that there are palmate compound leaves, and everybody thought that they were, of one kind, palmate compound leaves. But really, palmate compound leaves can be divided into two very distinct classes. So here's one class where the adaxial domain runs down the rachis. The rachis is clearly dorsiventrally flattened. And here's a different kind, which you would see in Schefflera, where there is no adaxial domain running down the rachis. The rachis is, or petiole in this case, is completely radial, and leaflets are made in a 365-degree aspect, like spokes of an umbrella. This is also very clear at scanning EM levels. So this is what a non-peltate compound leaf looks like. It just makes leaflets, but then it doesn't grow to separate them apart, so they're bunched together. But then here is Schefflera, and you can see that the leaflets are made all around this central domain. So we were able to define two classes within these palmate compound leaves. I'm sorry, I was going backwards. Didn't mean to do that. And so then I want to describe one more leaflet-inducing factor and fold auxin into the seeds. So auxin is involved in the development of leaf complexity, and this was work done by Dan Koenig in my lab. We knew that auxin foci give rise to leaf primordia, and the regular spacing of these foci determines phylotactic arrangement of leaves. But what Dan was able to show was that on a compound leaf primordium, like tomato, and Miltos showed the same thing in cardamom, auxin foci determine where leaflets are going to form. Not only that, existing auxin foci have to be separated by growth before new foci can form. So some growth has to happen before the next set of leaflet primordia will arise. So we looked at a tomato mutation called lyrate, and showed that it expresses exactly in these regions where the auxin foci would be, and mutating lyrate actually reduces leaf complexity quite a bit. So we said that auxin and lyrate are acting as these focal points, and we already know that NAM and CUC help to separate existing leaflet primordia. We also then did overexpression of this lyrate, which is the jagged ortholog in tomato, and showed that what is separated in the regular compound leaf can actually be folded together by ectopic braid outgrowth. And this was work again done by Rakefet, who was a postdoc in my lab, Dan Koenig, and others. Dan also showed, as part of this paper, that the reason we don't have these things coming together normally is because the tomato entire locus, which codes for an auxin response factor, actually suppresses auxin response in the various regions. And this prevents all the blades from folding into each other. So then to circle back to this idea of, what is the difference between simple and compound leaves, I alluded to this before, that, the role of indeterminacy genes suggests that compound leaves are only partially determinate structures. And then I showed you that several genes that actually regulate various aspects of blade outgrowth are involved in the process of making compound leaves, suggesting that this is not an either-or scenario. So with that, I want to then move on to the regulation of heterophylly, because in the end I want to tell you how these two programs come together, the heterophylly and heteroblasty programs. So heterophylly is the change in leaf shape, complexity in response to the environment. And in tomato we have particularly looked at it in response to shade or light quality. But perhaps the most dramatic examples of heterophylly are seen in the aquatic plants. So here in Roripa aquatica, grown in dry habitats, Roripa makes completely simple leaves, and if you submerge it, you get these incredibly compound feathery leaves. This plant really is amazing because it can respond very similarly to temperature, to light quality, and to various other hormones, and switch leaf morphology, and it can do it on the same sheet apex. So work done in Seisuke Kimura. So Seisuke Kimura was a postdoc in my lab, and he started to work on Roripa, and then he took this work to Japan, to Kyoto, where he has his own lab. And so now this paper is published with a postdoc of his as lead. And so Hokuto showed that when you have this change in leaf morphology in response to the temperatures, then at the lower temperatures, when the leaves are complex, the Notch and STM is much more highly expressed than it is at the higher temperatures. In addition, he showed that this is accompanied by an increase in CUC expression and the presence of CUC as a fractionating factor between leaflet primordia. And this CUC expression is much lower in the high temperature leaf form, and also no expression can be seen at the in situ level. So this is just a reminder to you about CUC, which we have talked about before. So in tomato, we explored heterophylly in response to sun and shade conditions. And so when tomato is grown under constant sun conditions, the leaves are actually simpler and under constant shade conditions. And I want to emphasize that this is physiologically not photosynthetically damaging. So we're giving the plants the same amount of photosynthetically active radiation, but we're forcing them to believe they're in shade by supplementing far red light. So when plants see far red light, they think they're shaded, but actually photosynthetically they're fine. So when we supplement with far red and the plants think they're in constant shade, they actually make leaves which are larger and more complex. So Dan Chitwood was then a postdoc in my lab, and he wanted to find out what was happening in these early stages of leaf development in the meristem and in the first plastophron leaf primordium. And so he did laser capture microdissection, and he then looked at what the genes were that were expressed in these two stages. And of course, we came up with almost this dream team of genes that you would want to see, because all the Knox genes, the Wuschels, clavatas, they were all in the meristem fraction. And then things like that, regulate blade outgrowth. So AS2 is the Fantastica gene, and this is the Arabidopsis annotation. Lyrate or jagged, as I showed you, TCP, ZABs, these were all present in the leaf primordium. He also then did laser capture dissection on meristems that were grown in shade conditions. So these were grown in the sun, and then he did a shade LCM and said, well, what changes when you give these plants continuous shade? And really not much changed in the meristem, although meristem size increased, and that's an interesting aside that we need to chase up. So meristem size increased, but not much changed in gene expression. But what happened was that meristem-related genes, the clavatas, the Knoxes, the Wuschels, actually went up in the leaf primordium in response to shade. And we saw a significant change because of the numbers. This is actually quite statistically significant. A change in the number of leaflets that were made in shade that were higher. So then I want to tell you that, in tomato, shade caused more leaflets to be made. But then here we're looking in the Ripa aquatica. This is just light quantity. Light quantity is reduced, so if you reduce light quantity, you get fewer leaflets. And this is paralleled with Knox expression. So under low light, the Knox expression is reduced. And similar results are seen in response to shade, but that's not part of the paper that was published, but I'm telling you that. If you want to see the data at the end, I have some unpublished data on that. So we have this situation now where in response to shade or low light, Knox expression actually goes down, but then I'm telling you in tomato that Knox expression goes up and leaf complexity goes up. So the heterophilic response is not the same response. It's really dependent on the species, and it's a labile response, and it's utilizing these Knox genes. So I want you to keep this in your head as you start, as we start to move towards the heteroblasty situation. So let's start thinking about heteroblasty. So first off, heteroblasty then is this change in leaf shape and complexity that we see as the plant ages. And this is a very standard change that really just happens in almost every plant. It could be very truncated, the juvenile phase could be really short, or it could be really expanded, but almost every plant is going through this change. And then of course this heteroblastic change that goes from the juvenile to the adult vegetative morphology culminates then in the plant going into reproductive mode and flowering. And so when we want to think about factors contributing to variation in leaf complexity and leaflet shape or heteroblasty, then I want to then point out Scott's really nice work done in the acacia system. So in acacia, leaves start out complex and they start to go more simple. And what you see happening is that one particular microRNA, and for the non-molecular biologists in this audience, I would just say that microRNAs are small RNAs that then regulate the expression of other target genes. And we will talk about what the target genes for this particular microRNA are in just a second. So he described two different microRNAs, but I think that they're very, very similar, often hard to distinguish. So I'm for now and from now on just going to talk about microRNA 156. So 156, its levels go down with age. So this seems to really be some kind of a developmental cue that plants have. And then in that same paper, Scott said, well, in looking at this change from juvenile to adult morphology, what we really have to think about is this microRNA 156 and the fact that when this is present, you have low levels of its target, which is the SPL genes. And if you're not a molecular biologist, it's just a gene. If you care, later on we can discuss more with the genes. So microRNA 156 downregulates SPL levels And SPL itself actually then feeds into the flowering program so that the plant can flower. So as the plant ages and MIR156 goes down, SPL levels can then come up and through feeding into several different pathways lead to the expression of these floral regulators that cause the plant to flower. So that's sort of the bottom line. So MIR156 regulating SPL levels and SPL then flowers. But it's not just about MIR156. There's a lot going on, and this is where the interesting stuff begins. So in addition to this microRNA 156, there are protein-protein interactions that become quite important for the regulation of leaf morphology and reproductive development. So let's see what's going on. So here are the SPL genes, I told you about them, and they are the targets of MIR156. So in the juvenile phase, MIR156 is high and SPL is low. And SPL is coded level. As MIR156 starts to tick down, SPL levels start to go up. So this seems fine. But where do the protein-protein interactions come in? So there is this other group of genes, the TCP genes, which Lena and others have worked on a lot because they regulate floral zygomorphy, if you will. And so the TCP genes complex with the SPL gene. But they can also complex with these genes, which by now should be familiar to, and these are the cup-shaped cotyledon or cup genes. So these are the genes that are really involved in making leaves lobed or making leaves compound. When there's no SPL, the TCPs bind to the cups. So those are these blue, yellow, and blue-green blue-red and green-red complexes. And then as SPLs come up, the cups free up. And when the cups become free, they can actually lead to serrations and increase in leaf complexity. So the SPLs are really playing a very important part in freeing cup-shaped cotyledons up so that it can then cause fractionation of leaf primordium. What regulates MIR156 levels? So Scott's lab also published a paper in 2011, and they said that leaf number and sugars in the plant might be regulating this. So they just, if you progressively defoliate the plant, MIR156 levels stay up and the plant thinks it's still juvenile. So one idea could be that as the plant is maturing, making more leaves, and sugars do the same thing, and perhaps having more free sugars, this is a signal that it's time to go into the adult phase and from then on to the reproductive phase. So then let's think about the intersection between these two programs, because we know that in tomato and in almost every other organism, if we care to look, there is a change in leaf shape in response to environmental conditions, what we're calling heterophylly, but there's also a change in response to aging or the heteroblastic change. If the change is in the same direction, are the same genes involved? That's the question that we wanted to ask. So here in tomato, you can see that not only leaflet shapes, but leaf complexity change with age. So leaf 1, 2, 3, 4, things are getting more and more complex with age, which is different from acacia. And so the experiment that, again, Dan Chitwood did was he took the same leaf primordium, but from progressively older plants. So the same plastocron. He's looking at plastocron 4 or 5 leaf primordium, but from progressively older plants, so that plastocron represents leaves of different ages. And so this is the heteroblastic series in tomato, and you could ignore this, and we could just look at what's happening in the P4 leaf primordium. So he clustered, and he did RNA-seq, large-scale data, and then he clustered the genes, and he found one set of genes that sort of goes down with age, and these are the TCP genes, and another set of genes that go up with age, and these are the cup genes. This makes sense because the leaves are getting more complex as we're looking at older and older leaf primordia. And then we also saw that the leaves get more complex in response to shade, and the meristem type genes start to get overexpressed in this case. So this was data I showed you. So we were able to take these two pieces of data and intersect them, and literally the only thing in common between these two programs of all the known players is this goblet gene, which is the CUG gene of tomato. So this tells us that while leaf complexity is increasing in tomato in response to age, the Knox factors are not involved in that process. It's really other things that are feeding into CUG that are responsible. So then this is a summary of that work, which says that, when we look at indeterminacy and shade avoidance, we look at this, response in response to evolutionary forces. The Knox genes are playing a role in all of these changes in leaf complexity. Then when we come down here to the heteroblastic variation, this is regulated by the TCP and CUG genes, and it does not seem to be a Knox-related phenomenon. Now, many people, including Cindy, have looked at heteroblastic variation and shown that sugars are important, neuron-56 is important. We worked on this tropical tree, the Green Aviana, and we showed that the SPLs and TCPs are important, and the CUGs, that's NAC, which is important. In heterophily, it seems to be Knox-related. This Capsella work, I don't know if they specifically looked at Knox or not, so I'm going to hold off on talking too much about that. And so the bottom line is that in the case of heteroblasty, things end up at CUG by one means. In the case of heterophily or evolutionary changes, developmental changes, things also end up at CUG, but by completely different means. So this seems to be the convergence point, but also probably the lowest point at which you're physically starting to separate leaf primordia. By lowest, I mean hierarchically. So I just want to summarize what I told you, and say that I showed you that leaf complexity is a complex problem, and many genes seem to be involved. Complexity can change with age and in response to the environment. I showed you that because genes regulating indeterminacy in the vegetative or the reproductive shoot seem to play a role in compound leaf development, compound leaves are only partially determinate features, organs, and then genes that regulate leaf dissection also play a role. So we really are not looking at an either-or scenario. They are sitting in the middle where both kinds of programs are involved. When we look at leaf complexity gene networks, and I really didn't go into gene networks at all, but I did show you about the PTS module. We have published a paper on a gene network that centers around the PTS module. So I can really say that the PTS network module plays a key role in generating diversity in leaf complexity, but it is also acting through the Knox genes. I showed you that heterophily involves Knox genes. These seem to play a major role in this change in leaf complexity that you can see often on the same plant in response to different environmental cues, and that the CUG genes are important in this fractionation process. I showed you that in heteroblasty there does not seem to be a role for the Knox genes, but the CUG genes are important in this process. And again, that when we start to look at the intersection between these two programs of heteroblasty and heterophily, really everything seems to come down to CUG, which then I tend to think of as a hierarchically downstream factor. And then heteroblasty itself is a progression to reproductive maturity and probably not very labile, whereas heterophily is a labile phenomenon. And this might tell us something about the genes that are regulating these processes. So maybe Knox genes are being used for evolutionary changes, for developmental changes, for heterophilic changes, because it's possible to use many different environmental cues to change the expression of these genes, but that the heteroblastic program is not a labile program. It is actually a very fixed, one-directional program because it's important for the plant to go from juvenile to adult maturity so that then it can go into the reproductive phase. And with that, I'll end here and I'll take any questions you have.